10 facts on enzymes. This video is to help you focus your revision on this very important topic, so it's where you'd start if you want to write your own notes. But be aware that this video does not contain all of the details you need. You should watch the other videos on enzymes and you should use your textbook. Also, be mindful that there are many practicals connected with this topic and students often ignore them. These will be asked on section B of your paper, so it's really important that you do revise the practicals, and there are videos on those also. Fact number 1. Let's define what an enzyme is. It's a biological catalyst. We know that enzymes are all proteins, so they are these special proteins, and their job or their function is to speed up biochemical reactions, and they remain unchanged at the end of the reaction. Without enzymes, biochemical reactions simply wouldn't happen fast enough to sustain life, so they are hugely important. Fact number 2. Enzymes we know are proteins, and when we consider proteins we have to consider shape. Most enzymes are proteins that are folded into a globular shape, and on the surface of the enzyme is this uniquely shaped depression called the active site. The active site is key to how the enzyme catalyzes a particular reaction. Fact number 3, enzyme specificity. This means that a particular enzyme can only catalyze one type of reaction, and it's all to do with the shape of the active site. Each enzyme you know has a uniquely shaped active site, and this active site is complementary or only allows one type of molecule, substrate molecule, to bind with it, and it's for this reason that enzymes are said to be highly specific. Fact number 4. The induced fit model or theory explains how the active site of an enzyme sets up those perfect conditions for the reaction to proceed. So when the active site of an enzyme binds with the substrate molecule, it changes shape to better fit the substrate, and so can set up those conditions where it can twist or strain chemical bonds, and this makes the reaction proceed. At the end, the product is released and the enzyme is unchanged. This model replaces an older, more rigid model known as the lock and key theory. Fact number 5, just a handy way to go over what is actually happening in an enzyme catalyzed reaction. So you have your enzyme and your substrate, and they temporarily bind at the active site of the enzyme to form an enzyme substrate complex. The reaction proceeds, the enzyme is released unchanged, and you have your products at the end of the reaction. Fact number 6. Temperature is one factor which greatly influences enzyme-controlled reactions. When you increase the temperature, you increase the kinetic energy of the reactants, and so they collide more frequently. There are more molecular collisions. And if the molecules are colliding more often, the chances are that they'll do so in just the right way and with just the right amount of energy for a reaction to proceed. So increasing the temperature of an enzyme-controlled reaction does increase the rate of the reaction up to a point. So as you increase the temperature in an enzyme-controlled reaction, the rate of the reaction generally increases due to those increased molecular collisions, and this will continue to happen until the optimum temperature for that enzyme-controlled reaction is reached. This is the temperature at which that enzyme is catalyzing at its best or most efficient rate. Above this temperature, enzyme shape gets changed. The active site can no longer bind to the substrate, it can no longer catalyze, and rate declines. Fact number 7. pH is another factor that influences enzyme activity. Enzymes work best within narrow pH ranges. Within such ranges, there will be an optimum pH at which the enzyme works best. For example, in the stomach, those enzymes, those proteases, prefer an acidic environment and a very particular narrow range of pH, generally between 1 and 2. So enzymes work within this narrow pH range, and within this range will be an optimum pH, a pH at which that particular enzyme catalyzes at its best or most efficient rate. When you alter the pH too much above or below the optimum, the shape of the enzyme gets changed. The active site can no longer bind with the substrate, the enzyme is said to be denatured, and the rate of reaction will dramatically decline. Fact number 8. Enzymes are widely used in the bioprocessing industry to produce products such as alcohol, medicines, food flavorings, etc. In industry, enzymes are often immobilized. This means that they are trapped or fixed to some inert material. In the lab, we trapped or immobilized our enzymes in a gel. And the benefit of immobilizing an enzyme is that firstly, the enzyme can be reused. Secondly, you get a purer product. And thirdly, immobilizing an enzyme often stabilizes the enzyme. Fact 9. Plant enzymes work best within a temperature range of between 20 and 30 degrees Celsius. Human enzymes work best around 37 degrees Celsius. Fact number 10. You can generally tell the substrate that a particular enzyme acts upon by its name. So a lipase is an enzyme that works on or breaks down lipids. A protease is an enzyme that breaks down proteins. So that concludes 10 facts on enzymes. Remember, this video is just to focus your revision to help you start somewhere. Remember to go over those practicals. They're worth a lot of marks. Best of luck.